3 hours? Are our brains
No,
going to be?
I don't think
okay.
it should be 3 hours because we don't have anybody here and there's going to be fewer questions.
Right.
So the point of this entire conference is so that everyone can know how they're being misdiagnosed. So that the public can be empowered to know what is accurate and what is inaccurate in terms of Lyme diagnosis. And the reason that I'm doing this conference is because a lot of people out there are misdiagnosed with chronic fatigue syndrome or fibromyalgia, or what they call wastebasket diagnoses, or a psychiatric diagnosis, or depression, or some other crap. And that's the reason these are called wastebasket diagnoses, because you might as well just jump in the wastebasket and your life is over, because that's how much you're worth.
That way.
The medical community thinks that you are not worth their time, and you are just trash, and they don't want to bother with you because of the spin associated with the fraudulent testing for Lyme. So the topic of this conference is scientific validity. What is a valid scientific term? I'm going to try to empower the public to understand that there are specific rules to determining whether or not your test is valid. I have to also operate the camera, so I'm going to zoom to the board. The criteria for scientific validity. These are not my rules. These are FDA rules. Now, I'm going to prove to you how you can confirm that these are FDA rules. And they're rules that apply to all science. A valid scientific analytical method, and you all know I'm an analytical chemist, so I used to do this for a living. Every single method I worked on to develop had to meet these criteria. Is it accurate? Is it specific? Is your method precise? Do you get the same number every time? That means, is the equipment that you're using okay? It doesn't have problems. Ruggedness. Can you develop a method, give it to somebody else, and say, here, run this, and have them run it on their equipment according to your specifications, and they get the same number. Limit of detection or limit of quantitation. That means how low in your analytic concentration can you reliably detect the analyte in question. Say I develop a method for, and I brought these textbooks just so you can see that this really is what we do. These are Cropat for gas chromatography methods. There's a whole bunch of methods in there that are standard. How do I detect, well, this one is for water-soluble vitamins, nicotinic acid, pantothenic acid, folic, and biotin, and so forth. In other words, these are standard methods, and they've been validated, and they're published. And if you need a method and you're a chemist, you're like, oh, let me go get that book, let me get that reference for a method. And so, if I wanted to find out how many parts per million of one of those vitamins there are in the local pond, and I develop a method and I say I can detect 10 parts per million of vitamin B in such and such a pond, because I developed a specific method where, given a known quantity of that water, I can detect that very, 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 very low reliably, because I've spiked it, created a solution of pure water, spiked in that amount, and I detect it every single time. It's valid. The method's valid. And in the first handout, I did a search. You can see it's a Google search. And I typed in, you can see what I typed in is I circled scientific method validation, analytical chemistry, right? And I get 6 million, 7 million hits. And one of those hits is the FDA. The very first hit. See? You see it says guideline for submitting samples and analytical data for methods. So it's very, very common. It's common knowledge. You have to go through this procedure to claim that you have a valid scientific method for determining a quantity of something. And we have in our back of our mind is antibodies in a person's blood for Lyme disease that are specific to Lyme disease and detect nothing else and detect a very, very low concentration of it. In other words, how do we find out if we can validly find out if a person has Lyme? That's where we're going here. So there are these, in scientific validity, there are these rules that you have to abide by. And you cannot submit a method to go with your drug or anything else unless you've met their rules. And I picked another one for the next page in this handout. The purpose of method validation is to establish that the method is fit for the purpose. And on the next page, you can see specificity, a description of it. In other words, don't take it from me that these are the rules. Specificity, accuracy, repeatability, limit of detection, sensitivity, limit of quantitation, linearity, and range. What that means is, I'll just take the example of what we do at Pfizer. They'll give the drug development team will give us a drug. And they say, well, we're far along on this clinical trial, and here's a sloppy method that we've been using to quantitate it. We need it validated to go with the new drug application, or the NDA. And the United States Pharmacopoeia. Because every time you produce a drug, and it's out there, you have to have something listed for how to test it in the United States Pharmacopoeia. So that any chemist anywhere in the world, take your drug off the shelf, take it into their laboratory, and see if every single tablet has 100 mg, like it says it should. How do you do that? There's a validated method that goes with that, and it's in the USP. We develop it. And so, typical methods for quantitation of a pharmaceutical or a bioanalyte is chromatography. Chromatography literally means color writing. Another word for chromatography is separations science. I think I have a couple of chromatic minutes in here. You all have done the experiment where you've seen colors bleed on a paper. Or if you took this black magic marker on a paper towel and then stuck it in methanol or something like that, and it would expand out and it would separate into the different colors of the visual spectrum. You're separating out pigments, right? So each pigment is an identifiable species, right? In a magic marker, or if you went out and took a whole bunch of leaves from the same tree, ground them all up, spotted them on a piece of paper, and then stuck them in like methanol or something like that, and watched those species migrate up. You'd see the different colors, the different xanthines, and the different chlorophylls. They're different colors. There's more than one chlorophyll. You know what I mean? So what you're doing is separating out the individual species. Well, that's good. You know that. You know you can try different methods to kind of separate this junk you have in a mix. There's a whole lot of other means of separation. If someone were to give you a sample of a complete unknown, like this has happened to me in the laboratory, people just give me samples and say, Kathleen, we found this junk in the effluent lines at such and such a plant. What's in it? You know, and you open up, like, illegally do this. Oh, you know, and because that's just an instinct, right? Is this organic? Is this laboratory stuff, or is this, you know, what is this? You're not supposed to do that, but that's an instinct. You know, where do I start? And you would take samples of that unknown, several of them, and you would extract them in different solvents. You know, some things are water soluble, and something doesn't. Water is a polar solvent, because it dissolves salts. And then there are organic solvents like methanol or acetonitrile or hexanes like gasoline, and they'll dissolve different things. So the goal for analytical chemistry is to identify a species, and then you want to know how much of it's in there. And that's where we're going with this. Because once you've separated out a species and have found that your method doesn't identify any other underlying species, and by species I mean like this. Each one is not a specific one. I don't really think. No, you won't grab it.
Can you edit certain parts?
I don't want to have to. I can, but I don't want to have to. All right, never mind. I will edit something else in there. So what we're trying to do is identify, see if a person has Lyme disease. So we're eventually going to go to looking for antibodies in the blood. And is that antibody specific to Lyme? So here are the rules in page 3 of the first handout. Sensitivity was what I just talked about. Can you detect a low level of analyte? And specificity. This is very, very important to this talk because a person can have any number of infections that don't make them ill, but we know that Lyme disease can make people very, very ill. And they have a chronic fatigue, like chronic immune suppression condition, weakness partially due to incapacity of the red blood cells really to carry oxygen. That's due to the infection. And there are many other markers of disease, which I'll get into later. But specificity, I'm going to read it right from this manual now. Method validation, IV standards, it's a standard. Specificity involves the process of line selection and confirmation for the blah, 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 blah ICP MS measures that are not significant. A comparison of results using a straight calibration curve will give information concerning matrix effects, drift, stability, blah, blah, blah. Specificity means, is the antibody or the analyte in question only coming from the species in mind? In other words, if you have 41 kilodalton antibody, which is flagellin, is it due to a Lyme spirochete? Or is it due to a spirochete? Or is it due to some other flagellated organism like chlamydia or helicobacter or whatever? You can see from this graphic right here, this is really out of proportion, but a spirochete, a cross section of it, it's like this. That's if I cut it right there. See? Cross section. I'll have this flagellar bundle. There's typically around 11 flagellar filaments. And what this is through here. You know, the whole length of it, right? The spirochete is little more than a bundle of flagellin with tissue wrapped around it. And how it moves is expanding and contracting the flagellin, so it looks like it's going like a screw. So there's a whole lot of flagellin in there. And if your little macrophage guys come along and eat one of these things, it's going to be digesting and chewing up a lot of flagellin. So it's very, very likely you're going to have antibodies to flagellin. So what these guys did, what Yale University did rather, was to try to find out if your 41 kilodalton antibody is specific to flagellin, or is it to Borrelia rather, or does it come to some other organism? Come from some other organism? Is it specific to Borrelia burgdorferi? Since everybody has 41 who has Lyme, just about. Okay, this is a better description.
But they're saying that's not enough just to have that. Or they...
Right. They're discounting 41, which is the wrong thing. Okay, so this is more data on method validation. You can see over and over again it has the same criteria. Specificity, accuracy, are you identifying the species in question and not some other junk? Can you identify it reliably every single time? Is it sensitive? Can you detect a very, 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 very small quantity of it? Can you hand off this method to somebody else? Very, very strict rules. None of these rules were put in place for the detection of Lyme disease, and that was deliberate. Okay. Now, so one search query you can do would be to go to Google and see if there are better descriptions or other descriptions of the methods to validate a method, the criteria to validate a method. Because what you want to do when you leave here is understand what I'm saying and be able to apply that scientific validity theory to everything you run into from now on. Not even necessarily Lyme or any bullshit that you get from doctors, but in any application of some sort of a detector of any sort, like if you wanted to buy a radon detector or anything like that. You know, whoever built radon detectors to sell for in your home, they've met this. If you run into a word like a psychiatric term, like anxiety, or that person has anxiety, that person has depression, and so you have to say, okay, well, what do they say is depression? Well, it's this, that, and the other thing. And they also say post-traumatic stress disorder is this, that, and the other. And so you say to yourself, okay, how can you prove that in the laboratory? Can you do any brain imaging to show that? You know, you're judging a certain person's behavior, but you don't really know what the mental processes that are going on. You can guess, but unless you do a valid analysis of the person, a valid medical analysis of the person, and rule out other things going on, like blood flow changes or encephalitis or something like that, those terms are meaningless. That's where we're going. So the next handout shows the National Library of Medicine. I would like when everyone leaves here they should be able to use the National Library of Medicine to confirm everything I've said, and to also use this tool yourselves. Get familiar with it. So all you got to do is go to Google and type in National Library of Medicine. You'll get the link. It comes up. It gets this page. Looks like this. What you want is PubMed to click on. Go there, and then you get the query page. And you can see that I've done a search on Borrelia and brain. I'm going to sit down because I'm already tired. You can see that the search results for the query input in the National Library of Medicine for Borrelia and brain at present results in 203 references, 203 citations. On the same page, in the top left, you see the asterisk where I wrote taxonomy? You see where it says search PubMed? That's a query bar. You will later be putting in taxonomy to search for how they classify these species. And then you click on these links and get the abstract. Some of them have full texts. But you will discover that Borrelia has long been known to be a permanent brain infection. The next query that I did was regards Alan Steer and blood work. You see what I did? You see what I typed in? Yeah. If you type in Steer and IgM and IgG, the 27th citation will be the original CDC method that Alan Steer developed for Lyme, which I gave you a copy of next. In the next one, Klempner and MNPs. There are 5 references where Mark Klempner is an author in which he discusses matrix metalloproteinases, which are degrading enzymes. He found them in the spinal fluid of people who have Lyme. He found nerve degrading enzymes in the spinal fluid of people who have Lyme. Now, if you go to the second handout I gave you, I'm kind of doing this in the reverse order, but this says matrix metalloproteinases in the spinal fluid of people with Lyme. Yeah, that one. If you look to where I point, Mark Klintner says patients with neuroborreliosis and encephalopathy often have subtle neuropsychiatric illness with memory deficit, irritability, somnolence, occurring months to years after classic manifestations of Lyme. Such patients have developed cerebrospinal fluid abnormalities, blah, 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 blah. And he goes on to tell you how he develops this specific test for matrix metalloproteinase number 30 by ruling out that it occurs in any other illness. He evaluates people with brain tumors, multiple sclerosis, lupus, ALS, every kind of brain sign there is. And he says, these Lyme patients seem to have matrix metalloproteinase number 30. That's a marker for disease. And you see in the abstract, 80% of Lyme patients have this marker of a nerve-degrading enzyme.
So they should be looking for that.
Of course, that's a marker of disease. Rather than do this valid testing on people, they diagnose you as crazy. In 2000, this report was in 1998. In 2001, the same guy reported that there are no objective signs of central nervous system disease in chronic Lyme patients. So that was an outright lie. Okay. But you can find that out for yourself. Go Klintner, MMPs, you'll find the report. You can't get the full text of it, but it's on my website. Okay, now back to the taxonomy database. Borrelia are spirochetes in ticks or lice. There are other kinds of spirochetes which you know about, treponemes, leptospira, that's about it, right? Brachyspira, the one that causes swine dysentery. But Borrelia are the spirochetes that you find in these arachnids. Almost all of them are pathogenic. In fact, spirochetes are parasitic spirochetes. They're parasites. They don't behave like other bacteria. So if you go to the taxonomy database, you will find, and you type in Borrelia, see where I had starred this Fukunaga. The phylogenetic analysis of Borrelia species is based on the flagellin gene sequences and its application for molecular typing of Lyme disease Borrelia. They classify spirochetes. They classify these Borrelia spirochetes by the relatedness of their flagellin, because they're mostly flagellin, and the differences in the spirochetes are, the wavelength, and the length of the organism, and how amplitude. They classify these kind of spirochetes and their relatedness on the basis of flagellin. Spirochetes are flagellin with protoplasm wrapped around them. Okay, so everyone perceives them as, okay, spirochete, which kind? Borrelia, okay, how are they related? So they always, and this is in the taxonomy database, look at flagellin. So they know that much about these organisms. Next, you turn to the next page. I printed out my search results, and you can do this yourself at home. How many different kinds of Borrelia are here? 155 strains, okay? But you only have Lyme disease, according to these idiots, if you have arthritis in a knee, which is just plain a lie, because you just saw the matrix metalloproteinase study. That was spinal fluid. There are all these kinds of spirochetes. Many, many, many of them, strains start CA. Those are in California. Those are spirochetes, and those can infect humans and mammals. But you don't have Lyme disease according to these idiots unless you have, B31, which is just a lie. Now, I printed out the full report of Kanaga, so you can look at what the phylograms look like. I'm not even, actually, maybe I didn't do it. Yes, I did. Here are the phylograms. So they classify these spirochetes by their relatedness to other spirochetes. This is how they perceive them. And what I said at the bottom of this was, these people who are cited by the National Library of Medicine say, we have focused on one of these genes, the flagellin gene, because it encodes the endoflagellar protein peculiar to spirochetes, and its diversity is valuable for distinguishing Borrelia. If these guys say we don't have Lyme and Lyme is a knee disease, are looking for spirochetes in ticks, they know how to look. They know what DNA to look for.
So why not do it in people?
Right.
Yeah. They can find that we have, go get our ticks tested, and, they tell us whether
It will
has
be,
the
they
Lyme
know, they go out there, they mash up a bunch of ticks, they take primers or DNA code from sections of specific Borrelia that they get from, okay, they can order these things, and then they amplify them, try to amplify certain types in the tick to see if this tick is infected with what strains.
Well, I thought you can't, it's hard to find it in the blood. Meaning, what are they testing on humans?
They're lying.
So it isn't.
Don't believe a single thing you have heard about Lyme disease, especially from Yale or the Steer Camp or the CDC.
Well, what's her name? Whitaker says she's finding most of the cyst form in the blood, and she's finding it everywhere.
The fact is they don't want to find it.
Yeah.
There are spirochetes in the blood. The relapsing fever spirochetes, which are slightly different from Lyme spirochetes, because the Lyme spirochetes are more tissue invasive. They're more virulent. They make you sicker than the relapsing fever spirochetes because they penetrate tissues more easily, and there are fewer of them in the blood, but they're all related, and they can bind antibodies to Borrelia in your blood, no matter what they tell you is lying. Just assume whatever they say is a lie, because it is. On the next page is a patent, and if you look up here in the URL, the patent database is uspto.gov. If you want to look yourselves at these patents, which I highly recommend doing because they say things in patents that they don't say to the public. They make claims about things that you'd be amazed because you don't hear them in the regular literature or in the BS that you get from Yale University. This patent is patent 5932220, and the inventor name is Alan Barber. It's I want you to notice that the application for this patent was filed on May 8th in 1995. This patent is a test for the Borrelia lone stari, or master's disease.
Down in Florida, but they're saying it's down there.
For 10, 11, 12 years, they've had this test. They have known how to detect stari, the one in the amblyoma tick, or the dog tick, or lone star tick, I'm sorry. And all this time, but did we hear about it? Is this test available? No. They say that we don't know what you have disease down there. They still say it. All right, and here is the molecular, this is a publication from 1991 by Yale University. People's names who are on this publication are Robert Berland, Errol Fickrig, Daniel Rahn, the author of this book, or the editor of this book, which we're going to get to. It's a 1998 publication. Daniel Rahn, full of lies. John Harden, who now works at Pfizer, and Richard Flavell. Fickrig and Flavell are still at Yale. Fickrig and Flavell's names are on the patent that goes with this. But here's what they did. This is very important. Molecular characterization of the humoral response to the 41 kilodalton flagellar antigen of Borrelia burgdorferi, the Lyme disease agent. The publication date is 1991.
So it's mostly the earlier studies that are saying how serious Lyme is.
Everybody knew early on, almost everybody with Lyme, that's 41, has antibodies to flagellin. Right. So the logical thing to do was take all these patients and see if they had Borrelia anti-flagellin antibodies. Does it come from Borrelia? Or do they have syphilis? They were just fine-tuning a test that they knew was the way to diagnose Lyme. In 1991, on the third page of that, you see this bar? This bar represents the DNA code for flagellin. What they did was they took sections of it at a time. They really did splice out these sections of code and splice them into like E. coli or something to generate this protein. And then they extracted and purified this protein. And they took the blood of people who had Lyme disease, people who had syphilis, and say, did their antibodies match that? Or did they match that? So they tried to find a region of recombinant flagellin protein that was specific to Borrelia. Everybody who they knew had borreliosis, only those people had antibodies to that. Specificity. You got to read this thing. You got to read this. I tell everybody, please read this over and over and over again. This is critical. They have known how to identify people with Lyme disease since 1991, accurately, specifically, and in the abstract it says the earliest humoral, that means body-wise, response in patients infected with BB is directed against the spirochetes 41 flagellar antigen. That means the earliest antibody is now specific, and they found that 17 out of 18 patients who they knew had Lyme had that antibody. Boom. Meaning every criteria for scientific validity.
How did they know they had Lyme? Was it EM rash?
Well, they came in with an EM rash and they sampled it or whatever. He'll tell you in the report. So they've known how to diagnose Lyme with scientific validity, meeting the rules. 17 out of 18, if they had done a few more patients, they probably would have found that was 95% specific. That's a criteria. Usually a valid method is 95% accurate, rather. Going to hit 95% of the cases. It's 100% specific. And they could do 100% specific for all the other kinds of Borrelia if they wanted to. You know, from the taxonomy list. So there's no question they know how to diagnose Lyme. Early on, Alan Steer, when I finish going through this, I'll show you what Alan Steer said about people who have 41 before they developed this method. And as regards people, comparing people who have 41 but have syphilis. Okay, so they know how to diagnose Lyme, they know how to diagnose all the other borrelioses, they know how to diagnose southern Lyme or Lone Star Eye or whatever they're calling it. And on the following the last page of that scientific journal report is the patent for it. Fickrig and Flagel's Yale patent for the method I just showed you, and it's 618533. They patented it, and they applied for a patent for it December 10th, 1993. All this time, they've known how to identify Lyme. The next handout I have is Alan Steer's original method. Yes, is that next? It is.
I might have missed one.
This is antigens of Borrelia burgdorferi recognize certain Lyme disease, right? The date on this report is 1989, 86. Alan Steer, are you with me? You got it?
Got it. 1986. That's a long time ago.
Yes, it is. Alan Steer and Joseph Kraft at Yale University said, we've looked at the blood of people who have Lyme for months to years, and we found that when people who have chronic Lyme will have new IgM and new IgG antibodies appearing over time, because this is a relapsing fever Borrelia, and what it does is antigenic variation. The surface antigens, this entire class of organism, Borrelia, has surface antigens that change constantly. They shed them, or they bleb them off. And that is because your body has created specific antibodies to a type of them, but they just get rid of that one, they make a new one, and so your antibodies are no good. So Alan Steer in 1986 well described what the blood testing standard, or what the blood testing looks like in a Lyme person. They all have 41, but over time we'll see new IgM. IgM means a new antibody. Every time this one makes a new one, a new antigen that's slightly different from the last, you'll make a new antibody for it, and that new antibody is an IgM type. But
many doctors will say, okay, you must have gotten bitten again, because it's a new, Dr. Kessler said that to me.
They don't realize that. It's because of the big lie of Dearborn, where they say you can't have Lyme unless it's IgG, and then when you do have all IgG, they tell you, oh, well, it's a past infection, so you're not sick now. Even though they know she's looking for IgM. This is the original standard. This is the original CDC standard for how to diagnose Lyme. Look at the people's blood over time and see if they have new IgM. That's how it's fear. That's how they used to do it. It's the complete opposite now, huh?
That's how they can tell if you have chronic Lyme.
Right.
Yeah.
If it's the bug's still there.
Even though they say there is no chronic.
Yep. At the very last paragraph of this.
Is there any way you can have Lyme and then not have it ever again, not have chronic Lyme?
Yes, if the bugs are no longer virulent, they can be there, but they can really not cause disease because they've lost plasmids and they can't do too much damage. But almost all Lyme is permanent, chronic. Can't get rid of it.
You can hold it down.
You can get well.
Yeah.
You can get well, but they'll always be there, just like latent syphilis. The bugs are always going to be there, and the minute you're down from car accident, surgery, divorce, or whatever stress in your life.
It's like herpes, too.
Mm-hmm.
Or chickenpox. That
Right.
might come back as shingles.
Never goes away. Exactly. The last paragraph here of Alan Steer's report says, although the current study did not implicate a particular spirochetal antigen as important in the pathogenesis of the arthritis, now he's only looking at arthritis, but still it's the same deal. The appearance of new IgM response and the expansion of IgG late in the disease, and the lack of such responses in patients with just the infection, the topical infection alone, suggests that VB remains alive throughout the illness. Comparison with other infections down here. He's saying the immune response to T. pallidum, that's the syphilis spirochete, has similarities to that of Lyme disease. Similarly, in certain chronic parasitic infections, immunosuppression occurs in the illness. That's chronic Lyme, right there. It's a parasitic disease that causes immune suppression, and the illness is associated with that.
And the
Specific
antibody response is restricted to a few polypeptides. What does that mean?
Well, when you have chronic late neurologic Lyme disease, you're going to probably have 41. Probably have low antibody response to antigens that are not typically found in the arthritis kind, like heat shock proteins and things like that. If you're lucky, and they're lucky and they can identify it in you. And syphilis and Lyme, serodiagnostic tests are often false negative. However, in syphilis, the use of a flagellar-associated proteins to which an early IgM is directed, rather than a crude sonic at the spirochetes on the solid phase analysa, improved the sensitivity of the test. Although antibodies reactive against this antigen may be present in patients with relapsing fever or syphilis, these diseases can be distinguished clinically from Lyme disease. So a person comes in with a chronic fatiguing illness and they have 41 and they don't have syphilis, they have Lyme or a borreliosis. Boom. That's out steer. Now, at the end of this, I compare to 10 years later, 8 or 10 years later, the Dearborn bans. Do you see that they just about have thrown out all the IgM? And you can only use these IgG. So now it's the complete reverse.
They throw out what counts.
Yeah. They say 41 doesn't count and don't look at IgM, which couldn't be a bigger lie. At this Dearborn conference, and I have the full booklet of it, CDC sent out invitations to like 10 or 11 laboratories across the country and said, come and participate in the proceedings. And then they completely blew off all the recommendations of everyone who attended. Because it was just a farce of a conference. They had no intention of listening to anybody else. They were going to do it the steer way. Because this way only captures 10 to 20% of all Lyme cases. Max. And in late Lyme, this method only detects, according to Mark Klempner, 4%. Late treated Lyme disease will have Dearborn positive in 78 out of 1,800 patients. If you're lucky enough to have the rare condition of Lyme arthritis in the knee.
And they make it sound like that's the major...
That's what they say is the disease, because they say you can't have a disease unless you have inflammation in the knee.
He's a rheumatologist, so everything's a arthritic problem.
It couldn't be more stupid. These are nervous system infections. Everybody knows that. Neurosyphilis, neuroborreliosis. These doctors, John Q, Public MD, are idiots. Plain and simple. Next, Borrelia burgdorferi, infection of the brain. Is the brain a knee? No. Huh?
Is it that they don't know, so they're ignorant, or
It's
they do know and they don't want to
Some do and some don't.
It's a combination of somewhere, every single one of them is somewhere between ignorant and arrogant. They don't give a shit.
Or criminal.
Right, that's if they're not the criminals. You know,
Right.
At Yale University.
There's lots of doctors
They
that...
believe what they read.
Right.
They just believe what they read.
Yeah.
Doctors are taught in medical schools by Big Pharma. Big Pharma tells them what's a drug. And now Kaiser is training doctors at New York Medical College. How's that for quality education? You just saw yourselves how simple that was, right? There are rules for scientific validity. Yale University met them in 1991. They've known how to diagnose any kind of borreliosis since then. You can go to the National Library of Medicine and type in Borrelia and flagellin. I've shown you how. You can look back and find that there were many other people at the same time as Yale who came up with a similar method.
Europeans probably too, right?
Everything in that database,
So what?
everything in this database, somebody has a patent for the Borrelia DNA flagellin.
Huh.
See?
What was the turning point where, and what was the reason that people said we don't want to find this? Meaning,
Osprey.
when did that happen and why?
Osprey. They wanted to sell a vaccine, and they knew they could never have a vaccine for a relapsing fever or borreliosis, because the minute you make an antibody for a certain surface antigen,
It'll change.
right, pinches it off, blebs it off, the antibody's useless. Antibodies do not work.
and they want to make money on vaccines, so they.
It was one big lie. Ospa, Lymerix was one big lie. They changed the definition of the disease to an inflammatory arthritis in a knee that is a hypersensitivity reaction to OspA, outer surface protein A on the Lyme spirochete. Yet there are a bazillion other spirochetes, a lot of them don't have OspA. They still infect your brain and central nervous system, and they're still permanent infections, and they invade your tissue, and they capture your bodily functions because they're parasites, and that's what parasites do. So this is boilerberg dorsifly infection of the brain. And the date on this is 1990. And he says in the, I only printed out the first pages, but the rest of it's on my website. In the abstract it says, the plasmid contact of N40BR was different from that of the infecting strain, implying either a highly selective process during infection or DNA rearrangement in the organism in vivo. What that means is, in this test, they took strain N40, which they happened to get out of a person's spinal fluid somewhere on Long Island, and they infected non-human primates, I think, right, or mice? Yeah, mice are non-human primates. And they have known that Lyme is a permanent brain infection forever. This book is published in 1975. I like this book too. And as you can see, in 1975 it says the ability of Borrelia, especially tick-borne strains, to persist in the brain and eye during remission and after treatment with arsenic or with penicillin, even after apparent cure, is well known. It's well known. And there are several other publications like that on my website.
Now, okay, just a quick question. Why, if it was Limericks that's the reason they didn't want to find Lyme, why would that affect the European progress?
Because SmithKline is a European company.
So
they wanted to sell vaccines on the European continent too. SmithKline basically said, and Yale owned the patent for Limericks. SmithKline basically said, we're taking all spirochetal infections and all infections from tick-borne diseases, and we're patenting them, and we're going to make the vaccines. Edward McSweigan worked at the NIH, Department of the Navy, I'm sorry, in the mid-80s. We think he was solicited by SmithKline because he then trashed the Department of the Navy, made fun of them, sent this snotogram to Senator Barry Goldwater, basically telling the Department of the Navy, or telling these senators and congressmen that the Navy was too incompetent to develop vector-borne diseases, and that it should go to SmithKline and academia instead. Tick-borne diseases. The Navy was too incompetent to develop vaccines for vector-borne diseases. Did I say that wrong?
You said to develop the diseases.
That's
why I know what.
So you're telling me that no scientist at the time, like a biologist, chemist, infectious disease back at that time, that questioned that. That said, wait a minute, we could never get a vaccine for syphilis because
Alan Barber himself was the owner of the other OspA patent, said there can never be vaccines for
everybody.
That's why there are no vaccines for relapsing fever. There are no vaccines for trypanosomiasis or sleeping sickness.
So no other scientists that stood up and said this doesn't make any sense?
They did. They weren't listened to, or they were threatened.
Just like the Lyme doctors aren't listened to.
No, and the doctors don't have the patience to do this work or listen to us.
We've invited
Hypochondriac Club.
There was a conference that that was put on at Lawrence Memorial. We organized it, but it was a bunch of different speakers, and we gave invitations to the hospital to hand out, and no doctor. I told you this idea. No doctors came. Then there was Dr. Levine or Levin or something that was there handing out information.
We got a lot of people who come.
Contradicted.
What was the name of that?
I don't know exactly.
Levine?
Yeah.
Yeah, he's the one.
He's around the corner from BJCCAN. BJCCAN will diagnose everybody who walks in with an EM rash, and then when they've had their 30 days of doxy and they're not better, he sends them around the corner at Flanders Four Corners, which is Route 1 and 161 in East Lyme, around the corner.
More than one Levine.
Oh, okay.
Robert Levine, I think. And then he Levine diagnoses them all with fibromyalgia and then abuses them. Gives them a little Ambien or a little this, and after a while says, no. Just abuses them. There's a lot of them out there in that area. So Broderick-Dorfride infection of the brain. So they know so much about the infection of the brain that they know to look there from an infecting strain. And they notice that the plasmid content has changed. So you're going to have different antibodies. So not only are you going to have different IgM, or new and expanding IgM antibodies, they might not be the same ones that are present in tick mash.
Can I say something just to break in? A friend who has a son with OCD from Lyme, she took him, Yale was having a study on this, and they told her no, on OCD. He cannot enter the study because he has Lyme, and it would still be in his brain, and that would be what would be causing it or something.
Got a million such stories. I'll write them all down and get their names. They can sue. No. So it's well known that IgM, the appearance of new IgM antibodies means persisting infection. The bug's still there. But the problem is that when they collect ticks to get the spirochetes out of them to do a Western blot, right? You need to harvest these spirochetes and culture them and use them to mash them up and then compare to your own blood to do a separation to see if your blood has antibodies that match something in the tick or the spirochete. Spirochetes become host adapted. If we took 125 spirochetes or 575 or whatever, and infected 5 monkeys with them, and then harvested those spirochetes from the monkey's brains, it's going to have a different surface profile and produce different antibodies than the one in early tick mash. So you still can only rely on 41. And that was recognized in 1990. Late in disease, you're not going to have early disease antibodies. So all you can rely on is 41.
And there's no way you can have 41 and not have early.
Right. If it's specific 41. You can have it and not be ill. Some people don't suffer illness effects. Bugs have lost virulence, or they're just sturdy and they don't have
immune system.
Or people who have chronic Lyme have something else, like they get something else acquired. Whatever. You can have 41. The thing is, 41 in illness.
Oh, in illness. So if you just have 41, let's say 85% of the population in Connecticut have 41, but only the ill ones should be treated.
I would think.
So if you have 41, you're not ill.
But know that you have Lyme, because if something else happens down the road.
Yeah, right. We should all be tested. That should be an automatic test.
I think they have done some surveys like that. In Rhode Island, in South County, which is the most highly endemic Lyme disease area in the world. South County, Rhode Island, highest tick density. In that county, 8% of the people there have babesia.
8%?
8. So how many do you think have Lyme? At least half.
And this is all that they're saying. And who knows all the
No, that's a real data point.
I mean, not everybody.
They didn't say how many were positive for Lyme. That's the thing, because they don't want you to know how many people. It's approaching half the population or more in Rhode Island. Not all of them are sick. We concede that. You know.
Does treating a not sick person at 41 prevent a future or help in the future, or just leave it? Don't stir it up.
My opinion is, a person who has 41, if we can get this specific test, should always have in their medical records that they have Lyme. If something else happens down the line, they can get the treatment that they need. If their symptoms emerge, particularly central nervous system symptoms.
I don't think
What's happening now is they don't want anyone identified with Lyme, because if you get that in your record, you're entitled to ceftriaxone treatment if you need it. And ceftriaxone is $12,000 a month cost or whatever. These bad guys who are behind this scheme don't want anyone diagnosed with Lyme. That's why they're further narrowing the definition in the treatment protocol.
Is ceftriaxone generic?
Not yet.
What's the company that makes it?
Roach.
Wouldn't they be very interested in?
They've been harassed. I got you. Okay, this is a page on my website. It happens to be this page. Lyme is a permanent brain infection. Lyme borreliosis is a permanent brain infection. So on the left-hand side navigation bar, the second link, there are more reports on. There are more published scientific journal articles which demonstrate that it is well known that Lyme is a permanent brain infection. You can get them. And the National Institutes of Health say that it's an infectious disease of the central nervous system and it's chronic Lyme and it belongs in the category of neuro AIDS. And here is the famous Alan Barber, who owns more patents than anyone for Borrelia, and he's the one that said there really can't ever be a vaccine for borreliosis, yet he owns the patent for the first OSPE, which is the Immulyme vaccine. He says, and I quote, this is from his website, these tick-borne infections are notable for multiphasic antigenic variation through DNA recombination. Now, I just said DNA recombinations in the case of relapsing fever, the occurrence of chronic arthritis in the case of Lyme disease, and invasion and persistence in the brain in the case of both these diseases. There's absolute ton of information out there that shows that they know Lyme is a permanent brain infection. Not everybody has symptoms. Later on you will, but you should know that get that diagnosis of Lyme and get ceftriaxone available to you should you relapse. And don't deal with asshole doctors. Find a new one.
I don't know if IV is always the answer. I mean, I never had it, and
I had IV. I was terribly, terribly sick for 9 straight years. Fever of 99, swollen glands, couldn't move. Literally could not move at times on the floor, stuck to the floor. Couldn't even lift my head. I got IV after 9 years for 4 months, and I got 100% better. 100%.
It didn't relapse quicker.
It did relapse, but at least I know.
We got better.
Yeah, and that's what it took. And I didn't even believe it at the time that I was getting it. I thought I was being steamrolled again. I'm like, come on now. I've been on oral antibiotics for 3.5 years. You really think this is? Oh yeah. You really think this is going to work? I didn't believe it. I didn't believe it. That is why I'm here today. I wouldn't be here today if that hadn't happened. 100%.
So if you get it again, it's the ceftriaxone you need, or will it always be effective for you?
I would think, and I would hope that too much time hasn't gone by and too much damage hasn't happened in the interim. People like me, and I think that's most people with Lyme, need to have a diagnosis of Lyme. Diagnosis should be chronic Lyme because that's the only kind that there is. It's chronic. If it's a persistent brain infection, it's a chronic infection. When you relapse, you should be able to get that ceftriaxone whenever you need it. And I'm thinking most people with chronic Lyme, at least as sick as I was, 2 or 3 times a year you probably need it to stay functional for the rest of your life.
For 3 months, 4 months at a time.
Probably just a month at a time.
Month at a time.
if it's staying under control.
Right, if you've gotten recovery. I don't think there's any sense to be made for going out even as far as 6 months. If you don't recover in 4 months, it's time to work on something else. 4 to 6 months.
Recover completely.
As far as you're going to get. This 1 year of being on ceftriaxone doesn't make any sense.
Of course, there's always the co-infections that need to be
Right.
treated.
Yeah, you need to work on you need to look for something else going on. Some other therapies, some other immune-enhancing therapies, natural, medicines that even possibly like a convalescent home, a rest home establishment where you have no stress and you're out in the woods and they serve you your food and you sit there on your rocking chair and you've got nothing going on.
You'd never be able to do that.
No one. I don't know anybody who would.
Is there a reason for a protocol, first-time patient that has this ill, to go at least 3 months of a high-dose oral to see if it would work, or is it right away to the IV?
Are you saying that the doctors?
I'm just curious because of the resistance, a lot of it is, we don't want to initially put everybody on an IV.
If you're terribly sick with chronic fatiguing illness and you feel like you have the flu, it's that toxic weakness where you feel like you need to have magical powers because you're so weak you can't even lift your arm up to turn the light switch off. Absolutely put them on IV antibiotics for at least 3 months.
But
Boom.
I didn't have to have it. get to this point. So my point is that I think they know that also, that a lot of people, not everybody, can be helped to get to my point. Maybe not as quickly.
Well, from a cost perspective, if you're
Right.
just looking purely at softening up the hard-edged physician who says, there's no such thing as Lyme. Well, what if, instead of everybody we tried 3 months of the oral, which is very inexpensive, it's generic.
High dose.
Right.
Yeah.
And, then it would cut down their resistance to considering
You would
Lyme
think
as
if
a-
that's the only reason they're.
If you're trying to convince
Diagnosed.
your doctor that chronic Lyme is a real entity and that he should be kind to you and try these drugs anyway, you're wasting your time.
They don't listen at all.
It has to come.
Yeah.
It has to come from a major splash. It has to come from the recognition that this is fraud. It has to come from the media. It has to come from grassroots. I, at this point, would not bother trying to convince John Q. average public, MD.
Unless he was a very
I would.
open-minded.
Well, I wanted to understand what is your biggest issue? That's what I wanted to know. So when I first brought this to the doctor and said, here is a CDC positive Lyme test after having 2 negative, I want you to see this. The very first thing he said was, oh, I bet you went out and found a doctor for an IV in your arm, didn't you?
Who said that? Who?
You're not
I'm not gonna talk about it on film.
Oh, right.
But that's what he said. He found a doctor to do it. He said, in my circles,
Is that Gottschalk? Yeah, he needs to be sent to jail. He knows what's going on, that bastard.
my concern is, I want to know what it is. Is it the cost?
No, they don't, the American Lyme Disease Foundation and Yale University don't want it known what I've just told you about Lyme. Chronic central nervous system disease, low antibody response, and the vaccine was a total fraud. They don't want it known that it was total fraud. They don't want people like you being diagnosed with chronic central nervous system Lyme disease or chronic Lyme. Because the treatment is OSPE, and it invalidates their spin that you're only going to have Lyme in a knee. Gottschalk is a neurologist and a psychiatrist. He is a big hoe. He's a liar.
I also say that it has to do with drug company profits on symptomatic treatments. Because
Yeah,
all
where's
these-
Pfizer on this?
There's just as many profits in antibiotic treatment.
Let me do a timeout because I gotta have a timeout.